So today I'll be taking up with you one of the most important historic event of the past decade, that was DNA, the discovery. And so when we walk through the path of how DNA was discovered, we need to go back 100 years ahead. DNA was discovered in the year 1954, 53, 54. So moving back from that year to another decade, that was Gregor Mendel. So you have heard about Mendel. So what did Mendel do? In 1866, Mendel discovered that there are some discrete units, because when Mendel discovered these discrete units, he couldn't name them. He couldn't say what are these units, because at that time the biochemical knowledge was not there to him. So just by seeing the experiments of by seeing the experimental results on the pea plants which he did, he could just comment on two things: that there are some discrete units which are being passed from one parent to the other parent. When Mendel said this statement, mind it, Mendel did not know the concept of gene. So he also didn't know what genes were. So that was in 1866. 2 years after, it was from the first time that Frederick Griffith, he discovered something known as deoxyribonucleic acid from the nuclei of eukaryotic cell. So he isolated something, but again, he couldn't decipher the structure. He isolated something which further later was established to be deoxyribonucleic acid. Now, if we come to 1905, so this was a very important discovery by Phoebus Levine. He discovered the order of a nucleotide. So we all know now that DNA is nothing but a polynucleotide, that is a chain of nucleotides joined together. So he said that the basic unit of nucleotide consists of a sugar, a phosphate, and a nitrogen base. This nitrogen base can be either pyrimidine or purine. So this was his discovery in 1905. So as you can see, the output was the discovery of DNA, but the process or the event started long back, as almost as long as 100 years back. So as I was saying, that the discovery took place in 1853-54, but the process started almost 100 years back. Now let's come to Thomas Hunt Morgan, 1910. Very famous scientist in the field of geneticist. He is known almost as a father figure. He worked with this fly known as Drosophila, or the fruit fly. If you put a banana on a dining table and wait for some time, you will be able to see small flies coming and sitting on those bananas. So those are nothing but Drosophila. So he worked with these fruit flies. So in 1910, and he observed that, what was his observation? The genes are located on chromosomes. So he developed this concept that traits are inherited. That was what Mendel said. Levine said that nucleotide is made up of sugar, phosphate base, sugar, phosphate, and a nitrogenous base. Morgan in 1910 further stated that genes are located on something called chromosomes. So he observed the flies and he came to a very similar conclusion like this. Now coming to Griffith. This was also a very landmark experiment, and I think so you have read the experiment of Griffith in 1112. So Frederick Griffith in 1928 studied the effects of virulent bacteria versus non-virulent bacteria. So he injected two categories of bacteria into mice, and he saw that in case of the virulent bacteria, it was observed that the mice were dying. So there might be something is happening. In fact, when he did his experiment, if you look at the experiment in 1928, so this is a bacteria. This is the S bacteria. S means smooth form of the bacteria. See, virulent versus non-virulent. So this is how the smooth bacteria looks, and this is how the rough bacteria looks. Now, when we inject the smooth bacteria into the mice, when he did that, the mouse were dead. He injected the rough bacteria into the mice, and the mice were living. So automatically, as a scientist, he concluded that the live S bacteria were the deadly form and the live R bacteria were the harmless form. So this was his conclusion. Now something very interesting happened. What he did, he took this S bacteria and heated them. So this is known as heat killed. So when you heat killed a S bacteria and inject it into a mice, you see the mouse are living. That means the infective property of the S bacteria has somewhat been destroyed on heating. Now what he did, he mixed the live R bacteria with the heat killed S bacteria. If you look into the experiment, when you inject only the R bacteria, mouse live. When you inject only the S bacteria, the mouse also live. But when you inject a combination of this bacteria, he observed that the mouse are dying. How can this happen? This can only happen if something from the R bacteria got transferred into the S bacteria. So that was his conclusion, as you can see. A transforming material passed from the S bacteria to the R bacteria, making them deadly. He, however, this was his experiment. This is where he stopped. He, however, could not exactly identify what got passed from R bacteria, from a heat-killed S bacteria to R bacteria. But he stated that something obviously passed, as a result of which the heat-killed S bacteria became virulent. He named it as the transforming material. So this is the rough and this is the smooth bacteria. Now coming to the searching for the genetic material. So as you can see, Griffith postulated and proved by experiment that indeed something is passing from the R bacteria to the dead heat-killed S bacteria, making them infective again. But what is this? So the next question to the whole scientific community was, what is Griffith's transforming principle? What is the chemical nature of the principle? And that is when these three scientists did a landmark experiment: Oswald Avery, Colin MacLeod, and Maclyn McCarty. Avery, MacLeod, and McCarty's 1944 experiment with Pneumococcus bacteria, where they wanted to identify the transforming agent that Griffith had already said that is present and that is being transferred from one bacteria to another. And in this, so this is another picture of Avery, McCarty, and MacLeod. And in their experiment, they proved that the transforming principle Griffith was talking was nothing but DNA. So it was for the first time in 1944, after the landmark experiment, the scientists could conclusively say that yes, it is the DNA that is being transferred from one cell, from the mother cell to the daughter cell. And it is this DNA which is now acting as a molecule of inheritance. Now coming to the discovering the structure of DNA. So in 1944, it became very clearly established that of the number of biochemical molecules we observe in a cell, we have lipids, we have carbohydrates, we have proteins, we have ribonucleic acids, that is RNA, and we have deoxyribonucleic acids. It is the DNA which is acting as a hereditary genetic material. So if DNA is the material that is being inherited from a mother to a daughter, automatically the question that comes up in the scientific mind is, what is the structure of DNA? How is this being possible? So next came the race to discover the structure of DNA. It was a very interesting race. I'm not getting into the politics of the race. I'll be going over only the scientific aspects of the race, but that was also a very interesting race. In 1950, you must have heard his name, Erwin Chargall. He first time said that in the DNA structure, whatever the structure may be, the ratio of the purine sugar to the pyrimidine the ratio of the purine bases to the pyrimidine bases is same. So if we know that adenine and guanine, thymine and cytosine. So if we consider the sugars adenine and guanine, purine, and thymine and cytosine as pyrimidine, so the ratio of adenine and guanine to thymine and cytosine is same. So it bears a 1:1 ratio. That is, adenine to thymine is 1:1 and guanine to cytosine is 1:1. So what does this discovery say? This discovery says that there will be, if there are 100 A's, then there will be 100 T's. If there are 50 G's, then there will be 50 C's. So this is a ratio that is constant. So if we have a given number of adenine in the DNA structure, then there will be also a given number of thymine. If we have a given number of guanine, then we have a given number of cytosine. So it's a definite geometric ratio. There is no randomness in the presence of these bases. And then came the famous Chargall's rule, AT-CG, the ratio is 1:1. So as I said, so Erwin Chargall moved forward to tell us the ratio in which the nitrogen base is present. If you remember, in 1944, Avery, MacLeod and McCarty proved that indeed DNA is the genetic material. Erwin Chargall further proved how the bases are present in DNA. Now coming to another very famous experiment by Alfred Hershey and Martha Chase. They used bacteriophage viruses, and through their experiment, they confirmed that yes, just like MacLeod, McCarty and Avery had said, that DNA is the transforming principle. In the Griffith's experiment, they proved through a defined set of experiment that yes, DNA was the genetic material that were used by bacteriophages. Bacteriophages are nothing but viruses which can kill bacteria. So even viruses have DNA as genetic material, and they transfer this material into the bacteria, killing them. So they clearly proved again, so this was second time it was proved, that DNA is the hereditary material. So this was the experiment of Hershey and Chase experiment, where very simply what they had done, they took a bacteria and they grew the bacteria in radioactive, they took the virus. These are the viruses, the pink color you can see. And they grew the viruses in radioactive sulfur. As a result of it, sulfur is a component of protein molecule. So as you can see, when these phages were allowed to infect a bacterial cell, this is the phage DNA and this is the virus. We all know that a viral cell doesn't enter into the host cell, it only injects the DNA. So what they got? They saw that the empty shells had the sulfur radioactivity. Nothing was seen within the bacterial cells. That is, protein is not the hereditary material. Next, when they give the bacteriophages with radioactive 32P, phosphorus. Phosphorus, as you know, is an important component of DNA. In the bacteriophages DNA, the phosphoruses got inculcated. These bacteriophages infected the bacteria, and you could see the radioactive DNA within the bacterial cell. So when it was observed, it was seen that the DNA, radioactive DNA, which was initially present within the viruses, have been injected into the bacteria. So this proved that it is the DNA that is being injected into the bacterial cell, not the protein. And that is how they concluded that DNA is a genetic material which is carrying all the information required for development of fresh baby viruses. So this was in a nutshell Hershey Chase experiment. Now, so in 44, MacLeod, Avery and McCarty proved DNA as a genetic material. Hershey and Chase experiments further proved that yes, indeed, DNA is the genetic material. Now started the race to discover the structure. In this picture, I don't know how many of you have already seen these people or not. This is Linus Pauling, very famous scientist who discovered the structure of proteins, alpha helix of protein. This is Chargall. I've told you his contribution to the discovery of DNA. This is Maurice Wilkins. This is Rosalind Franklin. This is James Watson. He was 25. So when this thing started, he was at the age of 22, 23. And this is Crick, Francis Crick. So these were the famous 6 scientists who somehow or the other contributed to the discovery of DNA. She is the tragic lady of the discovery of DNA. Wilkins, Watson, and Crick were awarded Nobel Prize for the discovery of DNA in 1964. Tragically, Franklin had died in 1957 due to ovarian cancer. She's one of my role models. It's a very tragic story with her. Okay. Now, coming to Maurice Wilkins. Rosalind Franklin used to work with Maurice Wilkins in a laboratory. They worked together. And Rosalind Franklin was an excellent X-ray crystallography. What is that technique? In a very simplified term, you take a molecule, you crystallize it, you simply crystallize it. And you pass an X-ray over that molecule, and depending on how the total element, I would rather put it this way, you take an element, you crystallize it, you pass an X-ray on that element, and how the molecules are arranged on that element, based on that, the X-rays will be reflected from that element, and the X-rays will fall on a plate, and the plate will give a picture of how the molecules are arranged in that element. So this is how she used to crystallize DNA. She used to pass X-ray over DNA, and then she used to get these photographs of this X-ray crystallography of DNA. So this is how she used to get those photographs. And this is Wilkins, okay. And this is James Watson and Francis Crick. They were very much interested in Linus Pauling work. By then, Linus Pauling had already discovered the alpha helical structure of proteins. And they were very much inclined to the fact that how Pauling had discovered the protein structure by alpha helix. And again, they were working on a model as to how. See, by then they knew that DNA was a polynucleotide. By then they knew that A, T, and GC are present in the same ratio. And incidentally, Maurice Wilkins used to come and show some of the crystallographic photographs of Rosalind Franklin to them. So they used to see those photographs, and they started hypothesizing their thought processes. If you look into James Watson's history, he was a zoologist. But if you look into Francis Crick, he was a physicist. So when X-ray crystallographic photograph is shown to a person who knows physics, who has a thorough knowledge of physics, it becomes easier for that person to grasp what are the bonds, how are the bonds placed, how many chains might be involved. So Crick had this capacity. And so this is that famous structure of Watson and Crick, as you can see in the background. And this is, I was talking about Linus Pauling. And this was the, he had proposed the alpha helical structure of protein. Not only that, he also proposed a triple helical structure of protein. By now, all of you know that DNA is made up of 2 antiparallel chains. But when Pauling started his hypothesis, he said that DNA is made up of 3 chains. So it was in 1954. Very around that time, so everything that is happening is around 1952. Hershey Chase proposed DNA is the genetic material. So huge amount of activity started in this area around 53, 54. So these 2 years. Franklin was taking photographs, X-ray photographs in 1953-54. She was a brilliant photographer, X-ray crystallographic photographer. And as I said, in 40, he had discovered the alpha helical structure. So based on that, he hypothesized that DNA is made up of 3 chains, which Watson and Crick were having a problem because whatever they studied, it was, and whatever already Erwin Chargall had said, and whatever they saw from the pictures of Rosalind Franklin, somewhere those pictures and the thought process of Chargall did not match with the triple helical model of Pauling. So there was a mismatch going on. And so they were constantly trying to problem solve. And this is that famous photo 51. And this photograph was somewhat also obtained by Rosalind Franklin, but incidentally, this photograph was shown to Watson and Crick by Maurice Wilkins. And on seeing this particular photograph, as I said, Crick was a physicist, and both Crick first and Watson gradually realized that the DNA cannot be a triple helical structure, and they came to the conclusive proof that yes, DNA is a double helix. And in 1953 April, Nature, they write, it is here we propose the structure of DNA. It was in luck of Rosalind that Watson and Crick published their result a month before of what she was planning to do. So once Watson and Crick published their result in Nature in April 1953, after 2 to 3 months, Rosalind, along with a lot of photographs, further emphasized on the structure of DNA. But as you know, the first credit goes to the winner, who is the first to decipher the structure of DNA. So even though it was this picture which actually helped Watson and Crick to decipher the structure of DNA, it was Rosalind's ill luck, and it was thus that they are famously known as the discoverer of the DNA structure. And you people have all studied Watson and Crick's double helix. However, you people have not heard the name of Wilkins, because as I said, Rosalind and Wilkins were in one lab and Watson and Crick were in another lab. So when the Nobel was given, it was decided the Nobel will be given to all the 4. But incidentally, 1957, Rosalind Franklin dies. Nobel is not given posthumously. That is, once you are dead, Nobel is not given to you. So even though she was the most deserving lady for the Nobel for the double helix, but the Nobel ultimately went in 1964 to Watson, Crick, and Maurice Wilkins. So as I was telling you, this is a crystal. So when you throw an X-ray beam, the X-rays reflect and they give an image on the detector. Based on that image, and as again I said, Crick was a physicist, he gradually identified that the DNA is a double helix. This is how the crystallographic picture was drawn. This is that experiment, Franklin's experiment. As you can see, since this being a double helix, when the X-ray is passing, you can see an X pattern. That is how they confirmed that it couldn't be a triple helix. Pauling had said it was a triple helix. They negated that because they found that this definite X pattern on the film. And that is how they concluded that DNA is a double helix. And as I said, she's the tragic lady of DNA discovery. Her photographs provided evidence that DNA is made up of more than a single strand of nucleotide. It also provided evidence that it is basically double helical in nature. And Watson and Crick, using her photo, seeing her photo, using the Chargall rule, finally came up to the conclusion that this was a double helix. This is a very famous picture of Watson and Crick. As you can see, Watson was very young then, 25. Crick was 10 years senior to him, 35 years of age. And this is the Rosalind Franklin University of Medicine and Science, where everybody knows her story. She has been referred to as the dark lady or the tragic lady of DNA discovery. And in his biography, Watson also gives due credit to Franklin regarding her discovery, her contribution. And finally, so the DNA discovery was something. We came to know the structure of the molecule that was considered as a hereditary unit long back in 1866 by Mendel. But what did it happen? What happened was that after the DNA discovery, that was the start. The subject of molecular biology started. It was with the discovery of DNA, the molecules within the cell became very important. People wanted to know how simple 4 letters A, T, G, C could carry informations that can give life, that can give characters, that can give traits. And Crick further proposed that how the information flows. It was proposed by Crick. He proposed that the information moves from DNA to RNA and from RNA to protein. And he coined this process of information as central dogma. So the central dogma hypothesis was postulated by Crick around 1955, where he said that information flows from DNA to RNA to protein. And this is the central dogma. What is central dogma mean? It means belief. So when you enter the subject of molecular biology, you have to believe that the genetic material is the DNA, which is true, and the information is percolating from DNA to RNA, which is also true, and from RNA to protein. Later on, in around 1980s, another group of famous scientists, David Baltimore, he proposed that this portion is not unidirectional. Initially, when Crick had proposed, this was unidirectional. He proposed that no, there are viruses who have RNA as the genetic material. So, in those cases, from the RNA, our DNA is synthesized. So, in here, we can see that the information is becoming bidirectional. So, if DNA is the genetic material, RNA will take the information. If the RNA is the genetic material, then on that RNA, our DNA will be synthesized. That is known as a complementary DNA. So this was known as revised central dogma. This was the new form of central dogma, where this process became bidirectional from DNA to RNA and RNA to DNA. So the information can be transferred from DNA to RNA and from RNA to DNA. However, the discovery of DNA, as I said, was for the first time helped in development of the subject of molecular biology, genetic engineering, huge amount of biotechnology. So that discovery in 1953-54 was what was, in case of bioscience, one of the landmark discovery. Just as landing of humans on moon was a landmark discovery, was a landmark achievement. It was not a discovery, it was a landmark achievement in the history of civilization of man. The discovery of DNA structure was a landmark achievement which paved ways for the subjects like molecular biology, genetic engineering, biotechnology. And not only that, once the structure of DNA became known, modern medicines also developed in a way that was not known to. So with this, I end my class here.